Hello everyone, welcome to Practically. We bring learning alive. And today I'm here to discuss with all of you the sample paper and its solution for class 12 term 2 exam for biology. So in the previous section we had discussed the sample paper 1, in that we had taken some important questions based on the chapters for term 2, and as we know that the term 2 exam according to the CBSE pattern is a subject to exam, so we need to practice and solve the questions accordingly, since the previous exam, the term 1 exams, was of MCQ pattern. So now since the pattern has changed, we need to have special practice for it. So here today I'm here to discuss with you all such important questions from the same chapters for term 2. We'll see the various questions, different sections, and then their solution. So I have already discussed the pattern in the last class. We have the 2-mark question, 3-mark question, and 1 5-mark question. So as we proceed, as per the question paper pattern, you'll also come to know. So let's begin. So we are going to begin with section A, and in section A, each question is of 2 marks. So we have total 6 questions in section A, and there can be internal options in it as well. So let's begin with the first question. Name the source of plant of heroin drug. How is it obtained from the plant? And it has, the question 1 has a part 2, second part also in it. Write the effects of heroin on human body. So since this is a 2-mark question, it carries 1 mark for each part of the question. So whoever watching this video, please try to answer. If you know the answer, you can write the important keywords in the chat box and let us know. If you have any query, that also please let us know. And then along with that, we'll also discuss the answers. So let's see the answer. So heroin, commonly known as smack, and its chemical name is diacetylmorphine. It is a compound. So obviously this is an additional information. I'm discussing this so that, because one of the aim of discussing sample paper is also to revise some important topics. So that's why the answers given over here, you will find some extra details in that. But while writing from exam point of view, we need to focus on the keywords. Okay, so that I'll be underlining. So that we need to pay more attention on. Okay, so heroin, commonly known as smack. So it is a white color odorless powdery substance which is obtained by the acetylation of morphine, which is extracted from latex of the poppy plant, and the scientific name of the poppy plant is Papaver somnifera. So we need to mention, since the first part of the question is name the source plant of the heroin drug. So the source plant of the heroin drug is the poppy plant, especially the latex of the poppy plant. It is more accurate, and also the scientific name we need to mention, that is Papaver somniferum. And I hope you all of you remember, while writing the scientific name, make sure you write like when, so this is printed, but when you're writing in the exam, make sure you underline the scientific name. The first letter should be capital of the genus and that of species should be small. Okay, so follow all these the rules which are given for writing the scientific names, and do remember the spellings as well. Since it is a written exam this time, we need to focus on spellings, especially the spellings of the keywords. Okay, so this answering this will yield you 1 mark. And the second part of question is write the effects of heroin on human body. So now heroin, since it belongs to the category of opioid drugs, and we know that opioid drugs, they bind or they show the effect on the central nervous system, CNS, and the gastrointestinal tract, which is also represented as GI. Okay, so they show effect on that, and they mainly act as depressant. Okay, that is, they slow down the body function. This is the main function of the heroin. This is how they affect a human body, and obviously overdose of it can lead to death, since it will hamper the function of the central nervous system and the gastrointestinal tract. Okay, so writing this part will yield you 1 mark. Okay, so this was our question 1 about the heroin drug, and this question is from Human Health and Disease, 2-mark question. Let's move on to the next question. Spirulina is a rich source of proteins. Mention few ways by which large-scale culturing of these microbes is possible. Okay, so this question is from microbes in human welfare, and we need to write two ways, like few ways, but two main ways, since it is a 2-mark question, how this can be cultured on large scale. Okay, so let's see the answer. So yeah, something about spirulina. So it is also called a single-celled protein, because spirulina is one such organism which is a unicellular organism, and from which we can obtain a huge amount of protein. And obviously, along with that, it is also rich in minerals, fats, carbohydrates, vitamins, etc. So these are the various ways where it can be cultured or grown on large scale. What are those? That is wastewater from potato processing, straw, molasses, animal manure, and sewage. So it also reduces the water pollution. So you can mention any of these different ways by which the spirulina, as you can see over here, the spirulina is grown mainly on the wastewater from potato and the molasses or the sewage. In this way, we can do the large scale cultivation. Okay, so writing the various ways will yield you 2 marks directly. Okay, moving on to the next question. Beta galactosidase enzyme is considered a better selectable marker. Justify the statement. Okay, so this question is from the chapter Biotechnology, Principles and Processes. So we know we need to use a selectable marker for differentiating between the recombinants and the non-recombinants, and there are various methods. The traditional method is what? Yes, it is by the antibiotic resistance gene, which is mainly we use the ampicillin and tetracycline. But beta galactosidase is also one of the method, but it is considered superior. Why? Okay, so let's see. So yeah, as I said, selectable markers is used in selection of recombinants from non-recombinants based on their ability to produce a color, okay, in presence of a chromogenic substrate. This is what the mechanism which beta galactosidase enzyme follows, okay, that it differentiates the recombinant from non-recombinants by producing a particular color in presence of chromogenic substrate, and the gene responsible for the enzyme beta galactosidase is actually an alternative selectable marker. So as you can see in this diagrammatic representation of a vector, so we have so the traditional way we have the resistant gene. For example, here it is the ampicillin resistant gene, and we also have the galactosidase beta galactosidase enzyme gene marker, where this is the lag Z region where we have this marker, and that will produce a chromogenic substrate. So how it happens is, whenever we introduce a foreign gene in this beta galactosidase gene, okay, then obviously that process is called insertional inactivation. Since if we insert a foreign gene in middle of another gene, that gene won't be able to code what it was originally coding for. So that's why it is called insertional inactivation. That gene will become inactive. And so then when it is grown on a chromogenic substrate, okay, the recombinants do not give colored colonies, right? Because the recombinant will have the gene, the foreign gene in it. So that's why it will not produce the enzyme, and hence it will not produce the color. The color also won't be produced. Okay, this is how this method works. So it do not give colored colonies, while the non-recombinants produce blue colonies. Okay, thus the recombinants can be easily visualized. Okay, fine. Let's move on ahead. List any 4 ways by which GMOs have been useful for enhanced crop output. So we know GMOs are the genetically modified crops. So what are the 4 ways by which this genetically modified crops can be useful for enhancing the crop output? So let's discuss. So first is, the GMOs, since they are made out of, we can add any useful trait in them. So the GMOs are mainly tolerant to the abiotic stresses. So abiotic stresses are the stresses caused by the abiotic factors, like we have the temperature and the water water inadequacy. So that's what, cold, drought, heat, etc. Okay. Then they also have reduced reliance on chemical pesticide. So the genetically modified crops do not need chemical pesticides because most of them have inherent properties to not let the insects eat them or harm them. Okay, that is what the pest resistant crops are. Then so we know, this is from the application of biotechnology. By using biotechnology, you can produce such plants which are resistant to the insects which cause harm to them. Then they have helped to reduce post-harvest losses, and then hence they increase the efficiency of mineral usage by plants. Okay, this prevents early fertility of soil. So it also doesn't let the soil become infertile by just one harvest. Okay, so in this way the genetically modified crops can be useful for enhanced crop output. Okay, so since they have asked 4 ways in this, so you will get half mark for each of the way. Okay, so if we mention all 4 of them, you will easily get 2 marks. Let's move ahead. Plants that inhabit a rainforest are not found in wetland. Explain why. Yeah, so let's see the answer for this question. So yeah, so the basic thing is rainforest plants, okay, why they are not found in wetland. So we need to understand that both rainforest and wetland are 2 different ecosystems. Okay, so they have the different factors in them. For example, rainforest have very high amount of rainfall over there. So basically the climatic condition, the ecosystem in all is different. Okay, so obviously the plants which inhabit rainforest are adapted to that particular habitat. And we know that the climate of rainforest is wet and humid. And in this climate, large trees and shrubs are predominantly present, because large trees and shrubs can easily grow in a place which has adequate water and humidity. Okay, so these plants are not adapted to survive in wetlands, because in wetlands the soil is deficient in oxygen. Okay, that is a property of the wetlands. They are deficient in oxygen, so obviously they cannot grow over there. So the plants develop above ground roots called pneumatophores or respiration. Okay, such an adaptation is not found in the roots of plants growing in rainforest, right? For example, here this is a wetland. Okay, and in this wetland you can see these plants, these are having these roots which are grown above the water so that they can respire, because inside water there is deficiency of oxygen. It cannot take up the oxygen the way they can take from the atmosphere. So this is a modification present only in the plants in the wetland. So the rainforest plants can't grow over here. Okay, it's a simple explanation. Let's move ahead. Differentiate between in situ and ex situ approaches for conserving biodiversity. So this is a chapter from biodiversity and its conservation. So let's see the difference between in situ and ex situ approaches. So here is a table which gives a difference. You can mention any 2 important points from this to differentiate between. Okay, so let's see first ex situ. So in this, the animals and plants are placed in a special care unit. In situ, in this, animals and plants are not placed in any special. Okay, because in situ is like for that particular habitat where they are present. Ex situ is we take out them from that habitat and then specially preserve them. Okay, so that's what, animals and plants are separated from their natural habitat. Whereas here the animals and plants are not separated from the natural habitat, but are protected in their natural habitat itself. Okay, then it helps in recovering populations or preventing their extinction under stimulated condition. Stimulated condition closely resemble to their natural habitat. So they make the habitat in such a way which will resemble to their original habitat. Okay, whereas here they help in recovering the population in surrounding where they have developed their distinct feature. So this is specially used for preventing the extinction. So when a particular species is like so less on a verge of extinction, then you will use ex situ. I mean, the ex situ way of conservation is used. But if that species is present, but you have to just increase the population, then in situ will be used. Then ex situ conservation includes protective maintenance of threatened species, and in situ includes protection of endangered species. So now we know these are the various terms. So endangered and threatened. So threatened is like little more worse than endangered. Threatened are like, you know, they are almost on verge of extinction. Endangered, who are less in number, they are not close to extinction, but they are less in number and alarming stage. So for that the in situ conservation is used. And the different examples of ex situ are zoological parks, botanical gardens, wildlife sanctuary, in vitro fertilization, etc. Whereas that of in situ conservation are biosphere reserves, national parks and sacred groves. Okay, let's move ahead. This is the next question. Now we are moving to section B. Okay, the next 6 questions are from section B, which are of 3 marks each. So let's see them. Let's see the first question from section B. Many plants and animal species are on the verge of their extinction because of loss of forest land by indiscriminate use by the humans. As a biology student, what method would you suggest along with its advantages that can protect such threatened species from getting extinct? So it's an interesting question. Those who are watching, can they think how can you use the knowledge of biology to protect these species? Let's see the answer. Yeah, so we can use the following methods. We have already discussed this in the previous question. Let's discuss this once again. We have first the ex situ conservation. Okay, that is threatened plant and animal species are removed from their habitats and kept in a special settings such as zoos, botanical garden, etc. What are the advantages? It protects the organism, it helps in maintaining their number and prevent them from becoming extinct. Okay, so this is one important method by which you can protect them. Then another method by it can be preserved. It can be also preserved by in situ as well. Then another method which can be produced by that gametes can be preserved by methods of cryopreservation. Okay, and can be fertilized in vitro followed by propagation by methods of tissue culture. So this is basically the use of biotechnology and use of science for protecting them. So especially if so for animal species we can preserve their gametes, and for plants there are something called as seed banks, okay, which are used for plant species. So basically the overall thing is we are trying to preserve their gametes, or in case of seed banks we are trying to preserve the embryos, okay, so that later whenever they are about to extinct they can be grown in sufficient numbers to prevent them from getting extinct. This method is called off-site method or method of conservation from outside the site. And advantages are what? Off-site collection may be used to restore the depleted population and reintroduce wildlife species and restore the degraded habitats as well. And it is useful in helping a large number of small area genotypes, okay, and rapid multiplication of even endangered species through tissue culture. So these are the various methods by which you can preserve the endangered species. Moving ahead, explain the exponential growth and logistic growth model of population. So yeah, so we know that there are 2 models of the population growth. One is the exponential and another one is the logistic growth. So for exponential growth, it happens only when the resources are unlimited, okay. So when the resources are unlimited, then the population will tend to grow in an exponential pattern. Okay, So how is the equation and the graph derived? So if the population size is n and the birth rate and the death rates are b and d respectively, then the increase or decrease of n, that is the rate at time period t, will be given by dn by dt is b minus d. So that is birth rate minus the death rate, how many are remaining, multiplied by n, which is the population size. So now if we consider b minus d is r, okay, so where r is the intrinsic rate of the natural increase, then the equation I am given as dn by dt is rn. Okay, r is the intrinsic rate of natural increase, and if we mathematically modify it by integrating, we can write it in this format. Okay, where n is a population density after nt is a population density after time t, n naught is the population density at time t is equals to 0, and r is the intrinsic rate of natural increase, and e is the natural base of algorithm, which like it's a mathematical equation, we use that. And here you can see is the graph. So if we plot the population density n against time, then you can see that this graph is exponentially increasing. So this is the basically the limit, but it's crossing that since the resources are unlimited. Okay. Then the second type of model is the logistic growth. Okay, and this happens when resources are limited. Okay, which leads to the competition between individuals and survival of fittest. Okay, so the population tends to grow logistically. So the population with limited resources initially, so how it will grow? Initially it will show a lag phase. So this is a lag phase where the growth is very slow. Then it will start growing very quickly. So this phase is called as exponential phase. Okay, and then finally it will reach a stage when the resources will be limited, the limiting capacity it will reach, it will become stationary or constant. This is known as, so this is exponential or also called as log phase, and then this will be called as stationary phase. Okay, so these are the three phases in which the logistic growth happens. Okay, so yeah, so whenever the population density reaches the carrying capacity, so the carrying capacity is represented by K over here, the carrying capacity of that particular area or habitat or ecosystem, whatever, wherever the population is present. This type of growth is also called as Verhulst-Pearl logistic growth. It's the name of the scientist who gave this model. And the equation is represented by dn by dt is rn. R is the same, that is intrinsic rate of natural resources, b minus d, birth rate minus the death rate, and n we know is the total population size, and k is the carrying capacity, k minus n upon k. So we need to remember and write this equation as well. Okay, so in this way you can explain the two models of growth, the exponential and the logistic. Okay, let's move to the next question. Mention the chemical nature of an antibody and name the type of cells they are produced by. Also write the difference between active and passive immune responses based on antibodies. So again, this is a question from Human Health and Disease. So let's see the answer. So yeah, chemically antibodies are nothing but proteins. Okay, and they belong to which family of proteins? Immunoglobulin family of proteins, which are Y-shaped large molecules produced by plasma cells, as you can see over here. This is the basic model structure diagram of the antibody. They are Y-shaped. So we have the heavy chain. So this particular chain, this is the heavy chain, and then followed with a small light chain. This is the light chain. Okay, and the above section is called as variable region, since they can change as per the immune response, the type of antigen which is going to bind. So this is the antigen binding site, as you can see, where a particular antigen with the epitope fits over here. And then this is a constant region which remains constant. Okay, it doesn't change. So this is the basic structure of an antibody. Okay, so they are produced by specialized white blood cells called the B lymphocytes or B cell. Whenever they get activated, they produce antibodies. And here is a difference between active and passive immune responses. Okay, so active response, it is a type of acquired immunity in which the body produces its own antibodies against the disease causing antigens, whereas passive immunity is also a type of acquired immunity in which ready-made antibodies are transferred from one individual to another individual. So in this, the own body is producing the antibodies, own antibodies, where in this the antibodies are transferred from the other individual. So this one has long lasting effect, but this does not have a long lasting effect since it is taken from some another individual. Then it is slow and it takes time for producing antibodies. This is fast and provides immediate relief. And examples are injecting microbes through vaccination. So if vaccination, most of us have got vaccinated recently for COVID, so that helps to introduce a body to that particular whatever substance which is present in that virus, so that our body is already ready. It starts producing the antibodies on its own, so it takes time for our body to be well equipped. But in case of passive, it is transfer of antibodies present in mother's milk, or also it can be transferred through plasma. Okay, so if you all remember, during COVID, many of the patients were given the plasma was transferred because they were the patients who already had got COVID, they were having the antibodies for it, and the other people who were more serious, they used to transfer the plasma so that they get those natural antibodies so that they can fight. Okay, so that is an example of passive immunity. Okay, moving on to the next question. What are some applications and advantages of stem cell technology? Okay, very wonderful advancement in the field of medical science. Stem cell technology, let's see. So stem cells are special human cells which are able to develop into different cell types, and this can range from muscle cells to brain cells. So they can develop into any type of cell of that particular organ, and in some cases they can also fix damaged tissues. And the stem cells are divided into mainly two forms. Okay, they are either embryonic stem cells or adult stem cells. Okay, and they are used for treating several clinical problems like tissue regeneration, damage myocardium after heart infarction, brain after stroke, and spinal cord after mechanical injury. So these are the various applications of stem cell. Tissue regeneration, if in any accident or anything, if some tissue is damaged, that tissue can be regenerated using those stem cells. Damage myocardium after heart infarction. So myocardium is the layer of the heart. So if that damaged part can be again regenerated with the help of these stem cells. Brain after a stroke. So during stroke, some of the brain cells or parts get damaged, that can be also recovered. And spinal cord after mechanical injury. So if there is mechanical injury in spinal cord, it can get bent or broken at some part. So there it can be regenerated using these stem cells. Yeah, so we have some advantages, more advantages. Adult stem cells have low rejection rate. Low rejection rate means if they are transplanted, our body doesn't reject it. Okay, it can be easily transplanted so that it can help the patient to grow. Stem cells having regenerative property can be used in replacing any organ which is not working or failing. Okay, that is obviously organ transplant is done. Instead of that, stem cells can be used to regenerate that entire organ and then transplant. And they can also be help studying human growth and cell development. They are extensively used in research, and they can also be used to test effects of medicinal drugs and medicine without use of animals. So in this way, the animal cruelty which is happening, that can be prevented using the stem cells. Next, name the two different categories of aerobic microbes naturally occurring in sewage water. Explain their role in cleaning sewage water into usable water. Okay, and here's the answer. So the two categories of microbes present in the sewage are bacteria and fungi. Okay, so these are the two main microbes present, and they grow and consume the organic matter extensively, which helps in reducing the BOD, that is the biological oxygen demand, and the COD, that is the chemical oxygen demand of the water, both of which are indicative of the pollution of the water. So if BOD and COD has decreased, the pollution level of the water will also decrease. So now what role they are present? Let's start with bacteria. So we know there are two types of bacteria, mainly aerobic and anaerobic. So aerobic bacteria will use oxygen, which is added mechanically into the wastewater, okay, which to break down the wastewater contaminants, converting it into energy. Okay, so and bacteria use this energy to grow and reproduce to help further cleaning the water. And anaerobic bacteria obtain oxygen from their food source, as anaerobic bacteria break down sludge, okay, and they produce methane gas. And you know, this methane gas can be used as alternative energy source in biogas, okay, since methane is one of the component of biogas, to fuel the wastewater treatment process. So it's like a self-sufficient process. That same methane octane can be used to fuel it, so that you know that system can get energy. And there are another type of bacteria called facultative bacteria, okay, which can switch between aerobic and anaerobic, okay, based on the environment. So they also help in some way. Then we have fungi. So fungi biomass can be helped for removal of following constituents. Okay, so certain fungi have ability to break down or transform organic material present in the sludge, and the most common role of fungi is consumer of organic matter in the system with low pH, means in the acidic pH. In acidic pH, bacterial growth is inhibited. Bacteria cannot grow in low pH or in acidic atmosphere. So there the fungi can grow and consume the organic matter. And fungi also have high enzymatic activity, which helps in degrading organic contaminants in the wastewater. Okay. Yes, next question. Study the diagram showing the entry of HIV into the human body and the processes that are followed, and identify A, B, C. So this is a diagram based question in which we have to study the diagram. So this is a question from Human Health and Disease, and the diagram of how HIV infects the cell. So there you have to identify what is A, like this cell, what is this cell, this A, then B and C. Where is C? Yeah, this is C. Okay, so let's see the answer. So A is the helper T cell. This cell, this entire cell which is getting infected, so the HIV enters which cell? The T helper cell. Okay, then B, this is the RNA, the viral RNA. This virus has injected its own RNA into the cell. And C is the enzyme by which it will convert it into the DNA. This is an RNA, and we know the human genome is nothing but DNA. So in order to incorporate into it, it needs to convert this RNA into DNA. That is done with the help of an enzyme called reverse transcriptase. So that is what this enzyme is, reverse transcriptase. Okay, so that is what ABC. You will get 1 mark each for answering these, and then you can easily score 3 marks. Okay, diagram based questions are very easy. For that, while studying, we need to pay attention on each and every diagram in our textbook. Let's move on to the section C, and this is the last question, which is of 5 marks. Okay, again very important section, very scoring one. Here's the question: what is population according to you? The size of population for any species is not a static parameter. Justify the statement with specific reference to fluctuations in the population density of a region in a given period of time. So let's solve this question. First, what is population according to you? So a simple definition of population is group of individuals living in a well-defined geographical area. Every word in this statement is important. Sharing or competing for similar resources, potentially interbreeding represents a population. This entire definition is very important. If you miss any word or any point in this, that will make the definition incomplete. So make sure you write every particular word in this definition. Group of individuals in a well-defined geographical area sharing and competing for similar resources, potentially inbreeding represents a population. Okay. Now, second part was, there is the statement, the size of population of any species is not a static parameter. Justify. Okay. So now the size of population obviously is not a static parameter because it depends on various factors. Okay. So that's what, depending on various factors including food availability, predation, pressure and weather. All these factors contribute to the population, and the density of the population in a given habitat during a given period fluctuates due to changes in 4 basic processes. So there are various factors, but these 4 basic things can alter the population size. What are those 4 factors? Let's see. Yeah, so here are those 4 factors. So we have the natality. This is the natality. Natality, which is represented by B. So natality refers to number of births during a given period in population that are added to the initial density. So basically it's like a birth rate. Okay, natality. Then mortality, represented by D, is nothing but a death rate, number of deaths in the population in a given period. Then we have immigration, is the number of individuals of the same species. Okay, this is very important keyword over here, of the same species that have come into the habitat from elsewhere during the time period of consideration. This is immigration, represented by I. And then we have emigration, which is the number of individuals of the population who left the habitat and gone elsewhere during the time period. Again, they need to be of same species as well. That is emigration, which is represented by E. Okay. So as seen in this diagram, all these 4 factors I, B, D and E, they affect the population density N. How? Natality and immigration is going to add up new individuals to the original population density. Hence they are represented by plus sign over here. Okay, because they will add up positively, increase the density. Whereas death rate and immigration will reduce the number of individuals and hence they are represented as minus. Okay, so these parameters keep on changing. So if there is very high natality compared to the mortality, then obviously the population density will increase. And if there is very high immigration compared to immigration, the population density will decrease. That's why it is not a static entity. Okay, and there is a formula also to give the population density after time t, that is t plus 1, that is original population density nt plus this formula B plus I, that is birth rate, natality plus immigration, minus D plus E, that is death rate and emigration. Okay, so in this way you can calculate the population density, and that's why it keeps on changing, hence it is not a static property. Okay, so yeah, this completes our 5-mark question. So this definition itself will carry 1.5 mark, and the entire definition or explanation will total give 3.5 mark. So in this way you can score 5 marks easily. So anyone has any doubt? If you have any doubt, even if you're watching this video later, you can leave us a comment, leave a comment on this video, and we'll be there to help you out. We are going to come up with more such sample papers. So we are done with 2 sample papers of biology, 1 more is remaining. We are also doing the same for chemistry and physics. So do check them out if you have missed the previous sample paper discussion. And after this, we'll also be coming up with more such things as the exams come closer. So stay tuned with us. If you have not subscribed to our channel, please subscribe practically 11th and 12th, since this channel is specially for 11th and 12th standard boards, NEET and JEE preparation. And if you have not downloaded Practically app, make sure to download the Practically app and register to start your free trial. We have many amazing simulations, videos, and all this content you will find on that app. So thank you for watching and see you next time.